Good afternoon. My name is Carol and I will be moderating this lecture today with my friend, Rosie. This broadcast is being carried out in a partnership with the Biological Control class from our entomology program, which is offered by Professors Angelo Palini and Madeleine Venzon. And this week, we are going to have one more lecture presented here as part of this partnership, so stick around. Today's theme is fungi and nematodes as pesticides. This is a topic of high relevance and has been gaining space more and more in entomological studies. And you are having the chance to talk with experts on the subject, so make sure your question is in the chat. As usual, today's broadcast will be divided in two moments. In the first moment, the guests will be freely addressing the subject in a presentation. And in the second moment, we will dedicate the space to the public, where the guests will answer the questions.
Welcome. My name is Jose. Carol and I will be mediating this broadcast. Biological pest control is one of the most studied insect control techniques in the world, which promotes reduction in the use of synthetic insecticides and favors sustainable food production. These benefits result from the hard-working researchers who constantly seek alternatives that promote a balance between production and environment safety. And today, Professor Patricia and Professor Sam are going to talk about this extremely relevant topic.
Now we have the honor to receive this great professor, Simon Elliott. He has a degree in biology from University of Southampton, master's degree in crop protection from University of Bristol, and PhD in agricultural entomology from Imperial College at Selwood Park. Professor Sam, now is with you.
Thank you. Good afternoon, everybody. I hope everyone is well in the pandemic. So we're going to talk today about fungi and nematodes as biopesticides. Fortunately, I had a PhD exam not too long ago, and I met Patricia, who will be talking after me. And she was part of the committee. She's going to talk about this subject, but I'm just going to give a brief introduction. I'm going to try and be brief because I think you want to hear what she has to say, someone from outside our institution. So we're talking about biopesticides, i.e. something to take the place, at least in some situations, of chemical pesticides. And here is an example, which is desert locusts. So these are from Africa and they breed in the Red Sea. And this is a fungus, Metarhizium, which I expect a lot of you have heard of. And these are locusts that I killed with Metarhizium a number of years ago. This is a very successful project that I'll just mention briefly, but if you want to ask questions afterwards, I can talk for about 5 hours about it. So each of these is a locust that is now sporulating and so is infective. And that's something I want to highlight briefly, is the potential using biopesticides. The one animal that you have reached and killed using a fungus or a nematode might serve as inoculum to infect another, which can be a good thing, but it can also be a problem, which I hope to mention briefly. So in the context, and I tried to give this context in the talk I gave on classical biological control, so here we're talking about inundative biological control. So the idea here is that we're really trying to put more of the natural enemy into the environment, enough our natural enemies into the environment to give control immediately or in the first generation without reproduction of the natural enemy. So I'm highlighting this as different from an inoculative strategy, which you might see, for example, using predatory mites, where you put some predatory mites in the field and you hope that the predatory mites will reproduce and it will be the subsequent generations that give you control. That's not what we're looking for here in principle. We're looking for what we put in the field to give our control immediately or soon. And that's a question as well with biological agents, that it may not be as quick as chemical agents. But then sometimes, like I just mentioned, you may have some secondary cycling. You may have further generations. And that's something we can mention and maybe discuss later. So this is the scheme that I put up when I talked about classical biological control, which is over here. And we're talking here about inundative control of biological control. So immediate control of the pest. And so we're doing this, usually we're talking about using the same techniques, application techniques as are used for chemical pesticides. So usually spraying, but then there are other possibilities, for example, granulated granules or other possibilities. So we're talking here about microbial pesticides or biopesticides, and we're talking here about them, they're used to control pest insects or pest arthropods, pest invertebrates. And then we can contrast this a bit with inoculative biological control, where we hope for population growth and subsequent control of the pest. So usually we're talking about predators or parasitoids here, but we may be talking about microbial agents as well. And we talked before about classical biological control, and there we have those first two steps, but we're also looking at permanent establishment. So that's something we're not talking about today. Focusing over here, inundative biological control. And then this question, if there is secondary cycling. So which combinations of organisms are we talking about? Well, we're talking about pest invertebrates. So I'm going to mention briefly locusts. Patricia will be talking about ticks. I don't know if she's going to talk about, I can't remember if she's talking about another pest invertebrate. And then here, for inundative biocontrol, we're basically talking about these microorganisms, with nematodes not so micro, but they're treated as microorganisms. Microsporidia are another group here, but they're in the fungi nowadays. So nematodes, fungi, bacteria, and viruses. And all of these are used for biological control, principally as biopesticides. So briefly, what are the advantages and disadvantages of biopesticides? We can consider bacteria and viruses in this, as well as fungi and nematodes. Advantages, specificity. This is very important if we're talking about environmental impact. Niche markets can sometimes allow that you label a product as organic, for example. And this is very important if we talk about, for example, production of organic beef in Australia. In Australia, they produce organic beef, and to control locusts, they can't use a chemical insecticide. So they use fungus, metarhizium. Secondary cycling can be an advantage, but it may not happen. When may it not happen? It may not happen when you've infected your insect or your invertebrate, and it becomes weak and gets predated. And this is what we see a lot with control of locusts. You can spray your fungus in the field, but you don't find any dead locusts because they all get predated. So you don't actually have a secondary cycling. Some advantages that you may not consider. One is that you may be able to produce these nationally. You may not be dependent on multinationals. So you may not be producing it in Switzerland or the USA or Japan. You may be able to produce it in Brazil. Or if we go over to Africa, you may be producing it in South Africa or in West Africa if we're talking about fungi for locust control. That obviously has benefits both politically and economically. Environmental impact, well, we're talking about biological control. You can't say there is no environmental impact, but it's going to be less than with chemical control. Advantages for the people operating, spraying the product, say, there's much less chance of health problems. And then you can, because it's just a different means of control, it can help you use a combination of strategies and maybe manage resistance to chemical insecticides, say. And that's a big advantage, for example, if you talk about control of mosquitoes. A lot of mosquitoes are resistant to chemical insecticides, and so biopesticides may come in and be very useful there. Disadvantages, well, specificity is a disadvantage again, because if you've got a product that just controls locusts, say, then you can't use it for other situations. So your market is smaller. This is a very important aspect. And again, niche markets. So you may not be able to cover those costs in such an economic way. Costs of producing the product, doing all the testing, all the safety testing, and then producing it as well. Secondary cycling can also be a disadvantage. Why is this? Well, if you consider that if you're saying to the public, well, we're producing a fungus or a bacterium, we're going to spray it in the field, and they'll say, well, is it going to establish? Is it going to infect other organisms? What's the environmental impact? Well, if you can say that it doesn't matter because it's going to kill the insects and then it will disappear because it's not reproducing, that can be an advantage. And indeed, there was a lot of early work done on viruses, for example, putting in toxins that made them kill hosts so quickly that they would not produce any more propagules of the virus. And the idea was that you could then sell it as more environmentally friendly. National production can be a disadvantage again. So I've put these on the same lines. Where you have a multinational, they can really get things done. Whereas if you're producing it at a smaller scale, it may be more difficult. But then on this aspect, for fungi, for example, a big benefit is that you can take, for example, a company that produces bread, yeast for bread. They may have big bio reactors, and you can just switch over production for a period to producing your fungus, for example, which can be very useful way to do it. And then shelf life. We're talking about biological organisms, so the time that you can keep the packet either in a warehouse or on farm, for example, is less than chemical products. So those are just some advantages and disadvantages. We can enter in more detail later if you wish. So I'm just going to mention briefly fungi. I've just got a couple of slides because I've not worked on nematodes. I've seen them, but not worked on them. So this is just a couple of examples. The fungi that you'll most see associated with or used as biopesticides are Metarhizium here. This is just found killing a leafcutter ant queen after the nuptial flight, and you can see the typical green coming out between the segments here. The spores are green, so the growth is white, but the spores are green. Then the other common one well-known one is Bavaria bassiana here. It's very cute, these photos. So those are the ones that are most used. There are other ones, for example, there's product being developed for whitefly control, Isaria, in Brasilia, which is very interesting. There are lots of other products as well, and lots of other fungi as well, but these are the main ones that are used. So this is just to show, especially if you've looked at viruses and bacteria that have to infect orally, usually, their hosts. Here we're talking about spores or conidia that will be picked up on the surface of the insect on its cuticle, and that will detect that it's on a certain, usually at the level of order, insect order, say, and it will produce a germ tube here, which will produce another structure called an aprosorium. This is the same with plant pathogenic fungi. And that's a structure that sits on the cuticle and then produces this thin peg, penetration peg, that together with enzymes exerts a massive force and manages to get through the cuticle. And then here in the endocuticle, it will reproduce and travel down, growing as fungi do, until it manages to get into the hemolymph. And here it will grow either as blastospores, which are the yeast phase, so don't have that fungal cell wall, which is important for how the host detects and deals with them, or they'll reproduce as hyphal bodies, which are basically small hyphae, small bits of hyphae, individual cells, and they have a cell wall, so that's different in the interaction. So here's the sequence. And they will often produce at this phase here, they'll be producing toxins which may debilitate the host, affect its immune defense, or kill it. And killing it is actually part of the strategy, because as you saw with the leafcutter ants, they have to emerge from the dead host. So they have to emerge from the dead host to sporulate and transmit to future hosts in a natural situation. So considering fungi as biopesticides, finding isolates is not a problem. The two pictures I showed you before, if you collect leafcutter ants during their nuptial flight and they're walking along the soil, you just collect them and keep them in a pot. Some of them will die and you'll be able to isolate Metarhizium and Bovaria. You can get lots of isolates doing that. Finding isolates is not a problem. And finding an isolate that will kill an insect. So this isolate here was found in soil, infecting a beetle, and here is infecting a leafcutter ant, so Bovaria. The main difficulties really are mass production, formulation of the product, so you can spray it, say, reaching the target in the field. So if you consider a pest that's on leaves in a field, it may be easier to reach than, say, if you consider assassin bugs, barberos, so vertebrate parasite hosts or vectors. They may be hard to find because they're not in groups, they're not at high densities. And then once you've managed to infect a host, the speed of kill may be a problem, especially if you're talking about a pest organism that is eating a crop now, and your biopesticide takes 15 days to kill it. That may be a problem. And then the secondary cycling may or may not occur. So I'll just show a couple of pictures of fungi against locusts. For some reason, there's animation here. So just to show this here, it's the same fungus on the same desert locust. So this fungus, Metarhizium flavoviridae, so it's a different species, it's a particular species, was developed to control desert locust. It was developed in an international project in the 80s. And it was found that this can be used against brown locusts. So these are brown locusts in South Africa. It's aggregation. This is a Dutch man, so he's very tall and he has to duck. I know I've had to do this as well, wearing a red suit, and you have to duck because you'll get hit by an aeroplane. And I was doing that when we were trying it against the red locust. This one's actually yellow, but it is a red locust. This one happens to be yellow. And these are all from Africa. And this fungus was produced and is now being used in Africa against the red locust. This was a project that I was involved in, and we went to Zambia and Tanzania, and we did field trials and showed that it could be used. So it's now substituting chemical pesticides, which is great because this is in wetlands. So it's very environmentally sensitive. So this is a product that can be used for different situations in Africa. If you can find the insects, which is not always easy in Africa, then you can use this fungus to control them. And the big advance with this program, a really big advance, was nothing so biological. It was actually the formulation in oil that allows it to be used in the desert. And the oil, well, if it's on the cuticle of the insect, provides the humidity for the spore to germinate and kill the insect. So it can be used in really hot situations and very effectively. So it's just mentioning a project I can talk about, or a situation I can talk about later. So I'm going to conclude my part, which is thankfully brief, thankfully for you. This is just a phylogeny from 2007, and this was about the period where, looking at this group of fungi, the Hypercreales and the Clavicipitales, so these groups, these ascomycete fungi, a lot of them are quite well known. So here's Metarhizium, here's Ophiocordyceps that infects zombie ants, there's the zombie ant fungus, and here's some endophytic fungi. Trichoderma is here somewhere, which is a parasite of fungi. And this just shows that during evolutionary time, these fungi have switched from from attacking plants to attacking fungi to attacking animals, again, insects. So a lot of them retain this capacity to infect plants, say. And this can be very interesting if you can use them as endophytic. You can inoculate seeds, say. So that's something I've mentioned here. This is a big area now, including in Brazil, which is using these fungi. So you can use Metarhizium, which has always been known as an insect pathogen. You can inoculate plants and you can get effects, not just insect control, but also growth promotion. So they have a lot of other effects. So this is very interesting. And then the market is growing and there are multinationals getting involved. So even biocontrol startups can get bought up by these big companies. So this is a common thing happening in Brazil, and there are a lot of jobs coming up in this area. So this is just a highlight to especially young scientists who may think this is all very beautiful, but will I get a job? Will I be able to do this in practice? So I'm going to finish now and pass you over, or pass you back so that Patricia has a bit of time to talk. Thank you for your attention, and I'll be back in a bit. Okay. Do I need to do something now?
Thank you, Professor Sam. Now, we have the honor to receive Professor Patricia Golo. She has a degree in veterinary medicine from the Federal Rural University of Rio de Janeiro, a master's degree in veterinary sciences, and a doctorate in veterinary science, both also from the UFRJ. Professor Patricia, now it's with you.
Thank you very much. So thank you to the Insectum group. This is a wonderful opportunity, and this will be fun and interesting being here. Talk to the audience about fungi and nematodes. Thank you, Professor Simon, for the invitation. I am really glad and I'm happy to accept the invitation when he said that, Patricia, could you talk about fungi as biopesticides? And I said, well, this will be fun. As he said, I am not an entomologist. I am a veterinarian. I hold a degree in veterinary, and also my master's and PhD degree is in veterinary sciences. So I'm a professor at the Federal Rural University of Rio de Janeiro, and also at the postgraduate course in veterinary sciences. So basically, I'm going to talk about the perspectives for biological control of ticks using fungi and nematodes. So my research line is mostly based on fungi, and every time that I give a talk, I want to give an overview of what you will receive and to reduce the anxiety, right? So this talk will be divided in two parts. The first part, I will talk about fungi, and the second part, nematodes. So I will give you a brief historical context of the use of fungi. I will talk about the why fungi are so popular and the main limitations. Some of them Simon has already talked, right? And so I'm going to give you some examples on how we can treat ticks. What are ticks and how can we treat them using bioproducts based on fungi and nematodes? And then I'm going to finish with some aspects about entomopathogenic nematodes. I don't have too much time, and I practice a lot to speak within 30 minutes. So if you have further questions, I'll be really happy to answer them. So let's start with our friend's entomopathogenic fungi. These are some pictures from one of my students, and she took beautiful pictures of these Meroisian isolates. As Simon said, when we say entomopathogenic fungi, the two genera that comes to our minds are Bovaria and Meroisian. These are pictures of Meroisian that were isolated from the soil here in Rio de Janeiro. To talk a little bit about history, and I'm really into history, especially invertebrate pathology history. This picture to the right is of Agostinho Bassi. I'm not sure if you're aware who Agostinho Bassi was. Actually, he preceded Louis Pasteur in the discovery that microorganisms can be the cause of disease. And something was going on with silkworms, and there was a white powder that was causing the death of these insects. And Lodge, that also known as Agostinho Bassi, just published this beautiful paper back in 1835. So actually, this paper describes the white muscardine, that is the disease caused by the fungus. And eventually, centuries after that, we have now the use of fungi to control arthropods, to control insect pests, and also to control arthropod pests, because ticks are not insects, right? I really like to say that. Well, to give you an idea of the market of microbial biopesticides in Brazil, I really like to show this picture from Gabriel Mascarin. It is published. It was published in 2019, and it gives you an idea that most of this market is so there are several products, but most of them are based on fungi. So you see 60% of the products in Brazilian market are based on fungi. And why are fungi so popular as biological control agents? Well, they are contact agents. So the arthropod doesn't need to ingest, to feed on them. So as Professor Simon explained, they just need to attach to the cuticle. They are really easy to apply, and they can be applied just like chemical products. So this is very interesting in the perspective of the producers, and they are easily mass produced. There are several companies in Brazil that can mass produce them. And this is a picture from Marcos Faria in São Paulo showing how Meroisium can be produced using rice, So there are actually several propagals that can be produced. So Areocanidia are one of these propagals, but blastospores, for example, can also be mass produced, and microsclerotia also can be mass produced. So we're going to talk a little bit more about these propagals and why are they nice. And finally, Meroisium and Bovaria, fungi, entomopathogenic fungi, are environmental friendly. So this is something that we really expect for as an alternative control, right? To change and reduce the use of chemical pesticides. So to talk a little bit about Brazilian historical trajectory in the biological control using fungi, I need to talk about Edmilson Marques and Dr. Pietro Guamiglione, which was a food and agriculture organization staff that arrived in Recife back in 1960s. So Brazil had several regional laboratories prepared for large-scale production of fungi, of entomopathogenic fungi. And these people started that. Also, Dr. Donald Roberts, my former PhD co-advisor, also was involved in the teaching, helping Embrapa staff and some universities on how to produce fungi. Why are they good for the control of insect pests? So this is a picture of Dr. Pietro Guaglione applying metarhizium to try to control spirobugs. So this is the first microbial control program in Brazil, metarhizium isopli versus spirobugs, and it's a very, very successful one and very famous one too. What is the current scenario of biopesticides registered in Brazil? Well, most of them are based on fungi, as I said. There are 24 products based on metarhizium and 13 based on Porphyria bassiana. So indeed, fungi are really popular as biocontrollers. Every time that we give a talk on fungi as biopesticides, there is a question. Are entomopathogenic fungi safe? So I decided to anticipate this question and bring these two papers from Zimmerman they were published in 2007. And the answer is yes. Mostly the use of fungi to control pests, arthropod pests, is safe. But there are some limitations, not everything is all good. And some of these disadvantages, Professor Simon said earlier for you guys, but I'm going to highlight one of them. And this is the tolerance of this fungus or of these fungi, when I'm talking about several genera, to abiotic effects. So what I mean is high solar irradiation, like UVA and UVB, can cause damage to the propagles, and they will not germinate. So low humidity also impairs fungal germination, as well as high or very low temperatures. So these abiotic effects will cause a negative impact to this fungi. So as I said in the beginning, I'm a veterinarian, and the research field that study the interactions between fungi and insects is really well known. But today I'm going to talk about the use of fungi and nematodes to control ticks. What are ticks? Ticks are arthropod pests. They are blood-sucking parasites, and they represent one of the biggest public health and veterinary problems in the world. So they cause anemia. They can cause anemia. They are vector-borne diseases, as I said. So they transmit pathogens that can cause diseases to humans and non-humans animals, right? And especially the cattle tick. So I work with Rhipicephalus microplus, which is known as the cattle tick, because the cattle are their preferred host. They can reduce the productivity of this cattle, so they can cause a huge economic impact. Not only Rhipicephalus microplus is important when we're going to talk about ticks, but also amblyomma. Amblyomma is the tick responsible for the transmission of rickettsia. And if you don't know, rickettsia is a bacteria that causes Brazilian spotted fever. So we are really surrounded by problems that are that arises from the tick parasiting hosts. These are some pictures of ticks parasiting bovine, ticks in the environment, and ticks parasiting dogs. If you don't know ticks, I'm pretty sure that these pictures will help you out. Ticks have an parasitic phase and a non-parasitic phase. And this knowledge will also help you find out the best strategy to control them. To give you guys some information about the dairy market in Brazil, so the importance of the dairy market, the dairy market in Brazil, according to data from IBGE from 2016, runs almost 40 billion Brazilian reais a year. And so the tick infestation can cause loss, several losses, economical losses. And so what do they use to control this tick? They use chemical acaricides. And how do they use them? Well, most of the time they do not use properly. They use inappropriately. And these can cause the emergency of resistant populations, resistant tick populations. So this is a paper from 2015, if I'm not wrong, and this shows how is the distribution of acaricide resistance in Rhipicephalus microplus in Brazil. So basically what I'm saying that this is pretty bad. So Patricia, you said fungi are already used for to control insect pests in agriculture, and now you're saying that they can be used to control ticks. And how does that work? Are there differences in arthropods' susceptibility to entomopathogenic fungi? And the answer is yes, there are. And also the strategy is different. When we're talking about insect, we apply the fungus to kill the insect itself and it's done. When we're talking about ticks, the tick female will die eventually after it lay their eggs, right? So what we want when we apply fungus or nematodes on the host or in the field to control ticks, we want their biological parameters to reduce. We want that the amount of eggs, the amount of larvae will be reduced after the application of this bioproduct. Okay, so Professor Simon said already how this fungus work and penetrate through the cuticle. This is a very nice picture that we have from some studies in our lab. And the picture shows the fungus killing the tick and coming out. So you can see the tick all green. And when we treat with Porphyria bassiana, it's all white. And when we treat with Metarhizium, it's all green. So it's beautiful, right? How can we use entomopathogenic fungi to control ticks? Well, there are two strategies, and one of them is the application on the host, and the other is the application in the soil, in the environment. And we've been conducting some research in our laboratory. The head of the laboratory is Professor Vânia Bittencourt. She's well known when we say when we want to say that about fungi and ticks. I believe she was the first one that started this research line in Brazil some decades ago, almost 3 decades ago. And so today I am part of her laboratory, and there are several studies that were conducted to apply the fungus on the host and also to apply the fungus on the field. And I'm going to show you guys one of them. This is a short communication from Mariana Guedes Camargo which she's now a postdoc at Vanya's lab. And this was a very nice publication that used a product that is registered for control of insects. You know, Metahill, the strain E9 and 9 from Coppert. So she used this product, and she used also oil in the formulation. And turns out she reported an average efficacy of more than 75% in comparison to the control group. So these are very nice results when we talk about a biological product to control ticks. Also, there are other studies that were conducted applying the fungus in the field. So this study of Mariana, they applied the fungus on the host. You can see here in the picture. And this study that was conducted by one of my master's student, Emily Mesquita. So she selected a native metarizon isolate. What do we think? Because of their limitations, the fungus can be very susceptible to negative environmental factors, right? So we selected a native isolate to try to compensate, because this isolate will be very familiar with the weather where the tick population is. And we had very, very good results when we reported the larval recovery. So we treated the grass, we put some tick females there, and then she counted the number of larvae that came out from these tick females, and it reduced it drastically. The same happened with another study conducted by Alain Marciano. And you can see here in these pictures, the tick female, and so it is covered by the fungus. You see the green here, and there's also some eggs here. So even though the female is dead and covered by fungus, it was able to still lay eggs. And this is something important when we talk about the control of ticks using fungi. It is because for insects, it's much faster in comparison to ticks. So it takes much more time to kill an tick if you compare to insects. And also, you need much more propagals, a higher concentration of fungi to kill ticks in comparison to insects. But we're trying to figure this out and to find best ways to reduce these traits. One of the questions, and one of the most important questions of this talk, is: are there products available in the market, in the Brazilian market, specifically for tick control? And the answer is no. So I've mentioned Metahill and Boveril, which are products that are available based on fungi, but to control insects. There are no products specifically for tick control in Brazil. And you can ask me why. And this is mostly because of the minimum criteria for tick product approval in Brazil. And it comes from the Ministry of Agriculture. So you see, it is needed at least 95% of average efficacy in stable tests, and 95% on days 7 and 14 after treatment for field tests. Remember that we reported 75%. So still, if the legislation didn't change, there's a long way to go until we have results that are compatible with the legislation. So if I keep talking about fungi, I'm going to keep, and I'm never going to talk about nematodes. So the second part of this talk, I'm going to briefly cover this subject. Well, during my PhD and my master's, I worked on fungi. I have some publications on nematodes, but these research in our lab is conducted basically by Professor Caio Monteiro and Professor Melissa. Professor Caio, that is a former student of Vânia, is now at the University of Federal University of Goiás, and Professor Melissa is from Federal Rural University of Rio de Janeiro. Okay, talking about nematodes. These agents are a little different from fungi. And this comes because they do not kill. Well, some of they can, but their virulence do not come from their action themselves. They have a relationship with bacteria. They own a bacteria in their guts. They have this bacteria into their guts, and the thing that will kill the insect, that will kill the tick, is the bacteria. So this is a picture from Anne Hayek and Jürgen Ellenbach book. And this is a picture from Professor Melissa that shows several infective juveniles and some adults as well. So these infective juveniles were produced in Galeria melanella, and you can see how the insect is really, dark red. So we know that it is infected by the entomopathogenic nematodes. So one picture that I really like, one publication that I really like, is this from Patricia's talk. And the title of the publication is Partners in Crime. And it describes perfectly the relationship between the entomopathogenic nematode and the bacteria. So basically, to make it short, the nematodes cannot reproduce without feeding on the bacteria that supply them with required nutrients. On the other hand, the nematodes provide transport and protection to the bacteria. So you see how they need each other. And okay, the use of entomopathogenic nematodes can be done basically as in two ways, using classical biological control. One example is the use of a nematode against wood wasp. And I got this picture from Larry Lacey publication, and also and mainly as an inundative release and augmentative releases. And for that, we need to mass produce Sternenema and Heterorhabdides. So Sternenema and Heterorhabdides are the main genera of entomopathogenic nematodes. And also, as fungi, they are also susceptible to negative environmental factors. Sorry. So to formulate nematodes, we can use vermiculite or other things that will make them more tolerant to these abiotic effects. I think I'm running out of time. We don't have commercial products in Brazil for tick control based on nematodes. But there are commercialized products indicated for tick control in the United States. And I decided to bring you pictures of Nematac. You see Sternenema and Heterorhabdides. One thing that is very interesting and should be mentioned is the association of entomopathogenic nematodes and entomopathogenic fungi to control ticks. Heterorhabdides is reported to be more virulent for ticks in comparison to Sternenema. And this picture here shows a tick that is dead because of the nematode infection. And you can associate the nematode, the entomopathogenic nematode, and the entomopathogenic fungi to try to kill the tick as fast as possible. And also, they say that the tick females are more susceptible to nematodes than fungi, and the immature stages of the tick are more susceptible to fungi. And probably that's because I didn't mention that, but I should. So the nematode infection, so the entomopathogenic nematode will use the natural openings of the insect, of the arthropod, to find the hemocell. But there are some nematodes like Heterohabdides that have some teeth, so they can actively penetrate to the host. So that's something that's important to mention, that worth mention. So I'd like to thank again the Insectum Group at Federal Rural University of Rio de Janeiro, Laboratório de Controle Microbiano. So this is not my research. This is research that is conducted there by these wonderful people. Please support women in science. Thanks to CNPq, CAPES, and FAPERJ. And also, if you like this talk, please support the channel, subscribe, and ring the bell. I think that's it.
Channel. Which channel? Qual canal?
Thank you, Professor Patricia. At this point, we will move on to questions asked by the public.
Okay.
Here's the first question for Professor Sam. Professor Elliott, a question from Angelo Palini. Professor Elliott, which is or could be the good candidate to control the Argentinian Brazilian Tropidacris schistosera recently threat?
Actually, good.
Excuse me, Professor.
Hello?
Your microphone.
My microphone. I haven't done anything, so can you hear me?
Yes.
Yes.
I was just saying that Angelo usually calls me Pentelho, so it's nice to get some respect. Good candidate is Metarhizium, of course, and this was produced. It was under development a number of years ago, so in the 1990s, by Embrapa in Brasilia to control grasshoppers and locusts in Brazil. Then what happened was that became soybean. And so on. So that slowed down, and there weren't big problems with locusts and grasshoppers in.
I'm sorry. We are having technical issues.
You bet.
Excuse me.
Hello?
Professor Sam.
Am I back? I think I'm back.
Yes.
Yes.
I can see myself on YouTube. Where was I? What did you hear?
I couldn't hear a thing, though.
Not
I think it
anything.
would be better if you repeat.
Nothing I said at all. Okay, so I first said that Angelo,
The next question to Professor Patricio. The question is to Bruno Franklin. Thanks for the presentation, Professor Sam and Professor Patricia. Can there be any antagonistic effects between the symbiotic bacteria of nematodes and fungi in a combined application?
Thank you for the question, Bruno. Actually, no. We conducted some research, and I think it's already published on using entomopathogenic nematodes and metarhizium to control Hypcephalus microplus. And we saw an addictive effect. You know, when we put bacteria, when we put the nematode and the fungus, we had better results in comparison to the fungus alone and the nematode alone. Could you hear me, guys? Okay, good.
Yes, thank you, Professor. Now, due to the time, we are going to our last question. Question for Professor Sam. Madeleine Venzon asks, Professor Patricia and Professor Simon, could you talk about the pros and cons of on-farm production of entomopathogens?
do you have any opinions, Patricia?
Farm production. What she...
There's the possibility of producing fungi
inside the...
It's done with viruses to some point. So
I'm not sure. Well, I have an opinion though, but I'm not sure if I have enough knowledge about this to conduct a discussion, but I'm going to share my opinion. And if you disagree, Sam, please feel free to talk about it. I'm not a big fan of producing entomopathogenic fungi when you do not have control about it. These agents, they can lose their virulence if they're not produced correctly. There are a bunch of technologies involved in the correct production of the mass production of fungi. So I'm talking about fungi because I don't have any experience with nematodes. But yeah, I wouldn't say that this would be a good idea.
Historically, there are cases of people, even factories, biological factories, producing aspergillus and selling it as metarhizium because of poor quality control. So if even biological factories can get wrong, then how can you do quality control on farm?
This is a big problem because, you know, the idea is very nice. Wow, producing fungi to control my arthropod pests, the arthropod pests that I have in my farm. But the thing is, there is a method to do this. You know, we are talking about a living thing. And there's virulent factors that is involved, also light, and it's not as easy as it seems.
And there may be human health risks as well if you're producing large amounts of fungi, for example.
Sure.
But then maybe at the level of a cooperative, it might be possible with the correct controls. And you may also consider not production, but scaling up you can collect viruses that have killed caterpillars and keep them until next year, and then spray caterpillars again, and then collect the actually producing the virus in the field, but you can do that. But then you may end up producing, you may get bacteria in that, and then it may not be so healthy.
Thank you, Professors Patricia and Sam. We had a lot of questions, but unfortunately, due to the time, we can't answer at all. But thank you for both brilliant participations. And now we are ending this afternoon's broadcast. We inform that this video will be available on our YouTube channel. And do not forget to fill the attendance list on the link in this video description.
Thank you, Carolina. Thank you, Insectum Group. Thank you, Sam.
We thank you for attending this event. Our lecture cycle will continue this Wednesday with Professor Richard Shaw. You're welcome to enjoy us, and you can get a certificate. Good afternoon, everyone. Wear the mask and take care.
Thank you, everybody. Thank you, Patricia, especially.